Yeah, I know, but I saw it.
Oh, yeah, that's right. You're right.
Have you ever been told of like they make you get
Yeah, I stayed for one person. People's choice.
उसका फायदा होगा बड़ा देखा
so you know that you have to
चलो बहुत अच्छा
and I got like 7 so we did like
मुझे आती नहीं
I'll send you
वो एक और बंदी मिली मेरा मेरा
he was in my I think he's in economics, so he's studying from economics.
All the best.
Oh, okay. We've already got this
Isn't that funny?
You're not supposed to know. If you write in such big letters, you know that. Just in case. Once a little bit proud.
Yep, I agree.
Wait for the fun.
I'll be like, we'll all just start singing.
ये मेरी should be वैसे वो ज़्यादा अच्छा कर सकते
We did send you.
Yeah, it's kind of hard for me to find stories that goes like
At least you can mention what I guess. Like, you know, that's what I don't think. I was also shouldn't go because two articles before
Oh yeah, every July. Denise. I pointed back to her and she would give some feedback. What they did well, what they need to work on.
इसके बाद
But I will, that's right.
Count for that.
So if you have questions,
Are the stickers aren't yet. They are, okay.
I was thinking about it, I don't know how much space I'm gonna get. Yeah, I have a space for it in here right here.
so, but I did on these two. And so when you get together later, put your heads together, if you have any ties, why, so what is the hope?
You can't decide.
Yeah, that'll be good. So, do you want us to move?
मैंने साइन अप इसलिए किया सर बाद में
Yeah, and they don't know
I'm gonna see if he might go first, Josh. So. Sure. And if so, we'll just mic him up. Sounds good.
फिर मैं अभी तो कुछ नहीं पहले मैं सोचता था
Yes, you won't be long.
Okay, that's fine. We'll just put a lot of light.
For the finalists.
Oh, sure.
Can you kind of help us? Josh and
We're gonna have them sitting.
oh, okay. Kind of in order then.
Well, I'll tell Nancy.
Oh, okay.
We'll try to make them in.
Okay, that way you'll know which one. So that way we'll cruise through it. Okay.
I think I got that.
Okay. So yeah, hopefully they'll just put them 1 through 10. So.
Everybody, I just have a quick announcement today. Today's a very big day in the graduate college, besides being the 3-minute thesis contest. It's also Dr. Graves' birthday. Everybody join us singing happy birthday for him?
No. No, really, seriously.
Seriously?
That's okay.
Anyway, we just want to say happy birthday, Bill.
Thank you.
Happy birthday. Happy birthday.
He's got cake outside, right? So he's brought a cake for him outside, right?
so it's not gonna be distracting.
Josh, I'll put your coat with mine.
microphones will appear on these screens. And if you have been selected, and if you want to text someone or email someone to let them know to look at our live stream website, you can do that. We won't start until 5 o'clock, so you'll have a few minutes to gain your composure and have a sip of water. Whenever you see your name appear, you'll be one of 10 in the list. We want you to sit in order in this front row on what I guess is the south side of the room. So could you arrange yourselves in the correct order? And as you get ready to come up and speak, there you go. Congratulations to the 10 of you. As you get ready to speak, when you're in the when you're queued up next, the team will come and put a microphone
Go ahead.
to get you ready to go.
Okay. Can we get this mic on you first here? You can put that just up on your...
And is this already on? Good to go. Okay. Thank you.
I have some extra slips for final streaming too.
Okay. Okay.
You might still be mic'd, I'm not sure.
Ready? So.
Okay, everything's on. Once you're done, I'm sitting over on the far end here, so just bring it back. Okay, good. It's already on. Everything's good. Okay.
If you'd like to read the release
Oh,
form.
thank you. Yeah, you're welcome. Thank you. Very nice. Very nice. Even says 3MT. Yeah, that's great. Yeah, if you could send that to me. I'll use it sometime at mags or something. Congratulations, guys. Yeah. Congratulations. serious time commitment. Yeah. Yeah. Okay. Congratulations.
Thank you.
So we're going to
Thanks
ask
for all your help.
the people speaking to primarily stay here rather than behind them.
And if we want to point to the screen, like, can we just stand, I don't know, long distance?
Yeah, and you'll be able to see the screen back there
Sure.
too.
Okay.
But you could say, on the upper left or
Sure.
on the lower right or whatever it
Sure.
is. Yeah.
Okay. Yeah. That's it. Thank you.
Or you can also walk over here.
I can?
Yeah.
Okay.
As long as you stay in front of everybody.
Okay. Perfect.
Yeah.
Okay. Thank you.
Yeah. Right there. Yep. Congratulations, everyone. You look nervous. That breath. Yeah. 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 Okay. Yeah. So we're asking, unless you feel really uncomfortable with it, we're asking everybody to stand in Well, welcome to the final round of the second annual Graduate College 3MT competition. I'm Bill Graves, Dean of the Graduate College, and I can tell you this is a really strong highlight of the year for us in the Graduate College at Iowa State, an opportunity to showcase the great research that our students are doing. And we thank you for joining us either here in person or via our live stream of the event. This final round comprises 10 graduate students, the top 2 from each of 5 preliminary heats that were held earlier this afternoon. The competition follows the protocols established by the University of Queensland, where 3MT began. Each speaker will be restricted to a maximum of 3 minutes and the use of one static slide that they designed. No questions from the audience are allowed, and the judges may not ask questions as well. We also request that the audience refrain from applause until all 10 speakers have completed their presentations. Out of respect for the presenters, we ask that you do not leave the room during the program, but if you feel you must leave, please do so between presenters. At this time, I'd like to ask everybody to check and make sure their phones are either turned off or completely silent. Before we get started, I wanted to take a moment to extend some thanks. First of all the student presenters. Earlier this afternoon, we had over 40 students compete in the preliminary rounds, and based on those that I was able to see, they did a great job. So even though we're going to be selecting winners here in the final round, I consider everyone who competed today a winner. No one was required to do this. The students all volunteered, and we really appreciate that they put themselves out in front of the audience and prepared their presentations. I also want to thank the 3MT organizing committee, 4 people who have been hard at work since August putting this event together. First of all, Denise Verkota, who I believe is hiding back in that corner, guarding the door. Judy Strand, who's at the other door. Katie Blair, and Sarah Huffman. They did an outstanding job working together and putting this event into place. I also want to thank the numerous faculty and staff volunteers who served as judges, room facilitators, timekeepers, and door helpers in the preliminary rounds. And last but certainly not least, I want to thank our 3 judges for the final round. Nancy Botker, who is a member of the Iowa Board of Regents for the state of Iowa, Ann Campbell, the mayor of Ames, and Kathy Obradovich, a columnist with the Des Moines Register. The judges will be deciding our winner and our second place speaker. The winner will receive $500 plus an all-expense paid trip to Grand Rapids, Michigan in April to represent the university in a regional competition. The second place finisher and the People's Choice winner will each receive $250. Everyone here and everyone remotely will be able to cast ballots on the People's Choice winner at the conclusion of the last talk. So let's get started. Presenters will be coming up to this area of the room in the order shown on this introductory slide, and I've asked them to stay roughly towards the center here in front of this table, and they will have a timer visible to them to keep track of time. They'll be using a slide that they designed that is intended to complement their presentation. So are we ready, Alex? Okay.
Hello, my name is Alex, and I have a dream to cure traumatic brain injuries, also known as TBIs. In the United States alone, over 50,000 people die from TBIs in a single year, and an additional 300,000 suffer from long-term consequences. A TBI is defined as an impairment to the brain's functionality caused by an external force to the skull, like an explosion or collision. Some examples of TBIs include concussions, post-traumatic stress disorder or PTSD, and Alzheimer's. Many of you have probably watched someone battle with one of these frightening injuries, but if not, let's look at the life of United States service veteran Daniel Summers. It is estimated that Daniel served in over 400 combat missions as a machine gunner in Iraq. Through these missions, he developed severe PTSD. His symptoms escalated quickly, and he is reported saying, quote, "My mind is a wasteland, filled with visions of incredible horror, unceasing depression, and crippling anxiety, even with all the medications that the doctors dare give," end quote. Shortly after this statement, Daniel took his own life at the age of 30. This is what traumatic brain injuries do. They have various symptoms that take away our most basic functionalities, and in the worst cases, they take away the gift of life itself while doctors are left scrambling for answers. My research focuses on studying the active mechanisms inside the skull of a TBI victim, and there's a big one. It turns out that the formation of microbubbles is the leading cause of TBIs. Let's use a football player as an example. When two athletes collide, there's an extreme pressure at the contact point of the skull, creating a low pressure at the opposite side of the skull where these microbubbles form. As the skull snaps back to original position, these microbubbles pop, sending shock waves to nearby neurons or brain cells. Some of you may be wondering how these shock waves from small microbubbles can lead to TBIs, but let's look at their significance in another application. It is known that microbubbles also form on the propeller of a boat. Over time, the formation and collapse of these microbubbles leads to significant wear on the steel foundation of the propeller. If microbubbles can wear away steel, then it is alarming to realize the damages they can have on vulnerable brain tissue. Our lab has developed a novel technique to create microbubbles that realistically resemble those in a TBI situation. We're collapsing these microbubbles on nearby neurons and analyzing the consequences. These simulations will provide an understanding and knowledge for pharmacists to design better treatments for TBIs. These simulations will also help engineers design helmets that absorb energy in a precise manner so that microbubbles will be prohibited from forming inside the skull altogether. TBIs have taken the lives of thousands of people and impacted the general quality of life in millions more. My plan, through my research, is to fight back and knock this problem in the head. Thank you.
My name is Carla Mann, and my thesis research focuses on using computers to predict relationships between RNAs and proteins, which can be used to identify new treatments for disease. But before we can understand RNA-protein interactions, we have to understand DNA. DNA is a complete set of blueprints for making any living thing, including humans. DNA blueprints are stored in a special part of the cell where the DNA can be protected, like how blueprints are stored in City Hall. But we still need to use these blueprints, so our body makes copies of the DNA, called RNA, and these RNA copies can be safely carried out of City Hall and used to make stuff. That stuff is proteins, which famously makes up our hair and muscles. But the human body actually produces over 20,000 different proteins, many of which have very important jobs like preventing the development of illness. Recently, we've discovered special RNAs that are never used to make proteins at all, but instead control when and where proteins are doing their jobs. Now imagine, if you will, if the construction crews around town got improper directions on when and where to do their jobs and did them at 3 a.m. in your backyard. Similarly, if there are disruptions in when and where proteins are doing their jobs, you can wind up with a whole host of diseases, including Alzheimer's and cancer. So if we know which RNAs and proteins in the body are supposed to interact with each other, we can then identify new targets for treating diseases that are caused by disruptions in these interactions. Now, if we were to try to do this experimentally in a lab, this could take years. However, we can use computers to speed up this process by identifying RNAs and proteins that are more likely to interact with each other and focusing our attention on these. This is where my research comes in. My research focuses on using computers to identify patterns in RNAs and proteins that we know interact with each other, and using those patterns to predict relationships in other RNAs and proteins, kind of like the eHarmony of biology. So, when we know that these things are interacting, we can get closer to solving all of these important problems in proteins and diseases. Now, my research contribution was to use this idea of protein disorder to improve interaction prediction. Proteins can come in highly flexible regions that are called disordered, and these disordered regions allow them to tightly bind to RNA. And so I used the idea of this protein disorder and incorporated it into my new prediction method called RPI disorder, which does about as well as previous predictions at predicting true RNA-protein interactions, indicated by a high true positive rate, but does much better at correctly identifying RNAs and proteins that don't interact, indicated by a low false positive rate. So my novel research of incorporating this idea of using protein disorder to improve predictions does indeed do just that, and brings us closer to a reality in which we can easily identify new targets for disease.
Good evening. My name is Jeba Jesdas. I am a parasitologist by training, and I'm going to talk to you about my favorite parasite this evening. Parasites are organisms that obtain their food and shelter from their host. Dogs have many parasites that live on them, and dogs have many parasites that live inside them. Of the most important of the internal parasites is the large roundworm of dogs, because it can also infect humans. Now, when adult dogs are infected, the worm can live on in the adult dog for a long period of time as the resting larva. The resting larva does not affect the adult dog, but when it is in an adult female dog, which becomes pregnant, these larvae that are sleeping can become active, awaken, move on through the placenta, and infect the developing puppies. Experts say that virtually all puppies are born infected. Now, in the puppy, which has just been born, these larvae move on and develop to become the adult worm, and these adult worms can lay up to 200,000 eggs per worm per day. Now, that is not a problem to the animal, but humans, especially children who pet these animals and who don't wash their hands, can accidentally swallow these eggs, which can initiate the human infection. Consequences in humans include loss of vision, abdominal pain, liver problems, and even neurological conditions. So then, instead of treating the disease in humans, it's easier to treat it in an animal, and that is where we encounter our first problem. We know that these larvae, even though the dewormer gets to these resting larvae in the adult dog, none of our dewormers that are available can kill them. And so my thesis started out by using this hypothesis that the dewormer is being pumped out as soon as it gets in, not allowing enough time for the dewormer to stay in the larvae and act. And so I described this pump protein, which kind of did what our hypothesis said it would do, in that as soon as the dewormer came in, the dewormer was being pumped out again. I was also able to show that by blocking this pump protein using a blocker molecule, we can keep the dewormer inside the larvae for long periods of time so that the dewormer can then kill the larvae. Now, this is important because the CDC says there are tens of millions of Americans who have been exposed to this larvae on a daily basis because people have animals and people pet their animals. And so my thesis contributes to One Health by preventing this important animal disease at the animal stage before it becomes a human disease. So for the sake of our children and for the vulnerable people among us who have impaired immune systems because of cancers and other diseases, we need to stop this disease at the animal level, and we cannot let these sleeping worms lie. Thank you.
Imagine one day when walking in the Iowa State University campus, you slipped and fell. You got a cut or scrape on your feet. Now imagine that this cut or scrape just wouldn't heal over weeks, even months. These are the type of chronic wounds that patients suffering from diabetes face on an almost everyday basis. Diabetes affects 1 out of 10 people here in the United States. It's a $25 billion cost on the annual health care in the United States itself. And millions of people across the world suffer from this disease. Unlike acute wounds that heal quite rapidly, diabetic patients, such as my own grandfather, had to be aware of even every single minute aspect of their daily routine, such as even shaving to not get nicks and cuts that could severely compromise their already delayed wound healing process. Conventional strategies for treating diabetes have largely focused on the monitoring of glucose levels in the blood. Only over the past few years has there been an interest in the use of natural materials for treatment of diabetes. My research focuses on the development of cheap and effective methods using natural materials for treatment of chronic wounds. When I create cheap hydrogels or jelly-like substances from natural sources such as algae or seaweed, we call them alginate hydrogels, we can load them up with medicines, apply them at the wound site, and at the very least, they'll form a protective barrier from infections from bacteria. What more they can do is deliver certain medicines and even cells to the wound site to boost the immune response of those suffering from diabetes. There has been a lot of research on how these wound healing treatments work efficiently. So one of the key methods in which we can do this is by looking at how the collagen is oriented in these kind of treatments. The way we do it at our lab is using a very sensitive technique known as second harmonic generation microscopy, which uses lasers without the use of dyes or any other expensive chemicals to visualize how collagen is aligned in these different types of tissues. By looking at how collagen changes from a very organized and aligned kind of an organization to one which is more chaotic and randomly distributed in a natural state, we can use collagen as a biomarker for understanding wound healing progression. Combining our knowledge of these gels and laser-based understanding of collagen, we can create effective treatments for chronic wounds in diabetic patients. All in all, it's my hope that someday all this work that I do on gels and lasers doesn't just become the background of a Bond villain origin story, but that which provides a brighter future for those suffering from chronic wounds from diabetes all across the world. Thank you.
One out of 68 infants born in the United States will later be diagnosed with autism. The rate of autism has increased 119% since the year 2000, making autism the fastest growing developmental disability in the United States. The earlier a child can be diagnosed, the sooner they can begin receiving services. Earlier services have translated to improved outcomes across the lifespan for people who have been diagnosed with autism. Researchers have identified early indicators of autism during infancy. However, most children are not diagnosed until closer to preschool age. All babies need one thing, and that is to be fed. Over half the women in the United States report breastfeeding their infant at 6 months of age. While the behaviors that an infant may exhibit during breastfeeding may seem like an easy process between mother and infant, those who have atypical behaviors associated with autism may face particular challenges. My research looked at maternal experiences breastfeeding infants who were later diagnosed with autism. The two most important findings of my study were that all the mothers had suspicions that there was something different about their baby, but they lacked the ability to clearly communicate it with their doctor. The second most important finding was that the majority of the participants actually used breastfeeding as more than just a source of nutrition. It became an intervention to calm, comfort, and soothe those behaviors that were different from other babies. If we shift the way we look at nutrition and move past just a way to feed our babies and consider it more like a milestone, the same way that we look at crawling or walking or talking, we may be able to identify and provide services sooner. My research hopes to use infant feeding as a way to identify, assess, and provide intervention to these infants and families so that we can begin to put the puzzle of autism together earlier. Thank you.
Water is life. All living things on Earth need water, and all living things on Earth are connected by water. The hydrological cycle, the global cycle that connects water in the atmosphere to water on the ground to the water that comes out of your tap, is vital to the health and sustainability of our environment. And yet, our cities dramatically disrupt that water cycle that is so vital to our survival. As landscapes change from natural environments to urban environments, roads, parking lots, buildings take over the landscape. These surfaces disconnect rainwater from the soil, leading to increased flooding as that rainwater has nowhere to go, leading to increased pollution as litter, de-icer on our sidewalks, fertilizer on our lawns gets washed directly into streams. Because when it comes to pollution, if it's on the ground, it's in our water. My research focuses on reducing the impact of cities on the environment by trying to reconnect that natural water cycle in cities. I first look at the human side of the problem because it's vitally important that we understand what people know about the environment, what they know about water, and what they know about their role in the water cycle. Because there are so many things that people can do in cities to help reconnect that natural water cycle, from installing rain gardens or rain barrels, to reducing the use of lawn fertilizer, or picking up pet waste. Along with the social side of my research also aims to quantify the physical problem by looking at the stormwater coming off of residential properties. I will monitor the effects of these small practices, the rain gardens and the rain barrels, to determine how much they help to reduce pollution and reduce flooding. I will combine them in a computer model that demonstrates at the city scale how big of an impact these small practices can have. Because cities aren't going anywhere. We humans thrive in urban environments. We just have to be sure that our success doesn't come at the expense of the natural environment. The biggest implication of my research is helping to increase the sustainability of our cities by engaging urban residents in the water cycle and bettering our understanding of the physical properties of stormwater. We can hope to move to a more sustainable future, one where our ecosystems are healthy, our water is clean, because water is life. Thank you.
Amyotrophic lateral sclerosis, more commonly referred to as Lou Gehrig's disease, is a debilitating neurodegenerative disorder that causes paralysis and ultimately death in 99% of patients. The majority of those affected have no good treatment option and therefore have no good hope for life after the diagnosis of this disease. I first became aware of ALS at the age of 13 when I lost my grandfather to it. His battle was a short 8 months, and I've been studying it ever since. Together with the Osdinler lab at Northwestern University, our research has been to understand the cells that are primarily affected in ALS, the motor neurons. These are cells that you have in your body. They're in your brain and your spinal cord, and their main role is to control movement by communicating with your muscles. In ALS, however, these cells degenerate, and they begin to withdraw from your muscles, leading to muscle deterioration. The majority of patients succumb to the disease within 2 to 5 years as a result of that muscle deterioration. Currently, there are 2 FDA-approved treatment options for ALS, which may sound promising. However, these treatment options help to delay the late-term symptoms of ALS and will prolong life a mere 2 to 3 months in those that they work for. For this reason, novel treatment options are necessary, preferably ones that halt the disease in its tracks. However, there is another large hurdle for researchers of ALS in that there are more than 50 genes and over 1,000 unique mutations that may cause the disease. That means that the cases may vary drastically, from patient to patient, and that their mutations can actually cause symptoms to appear differently. For example, one patient may notice that their right leg has not been working properly, whereas another will notice their symptom onset in their left arm, such as my grandfather did. As these mutations have been discovered, scientists will use them to generate animal models. Our work uses multiple mouse models of ALS, containing several different human mutations. We begin by isolating the motor neurons from the brains and spinal cords of the multiple models. We use both ALS and healthy mice. We then use next-generation sequencing in order to identify differentially expressed genes between the two populations of cells. The identification of genes that are more highly expressed in the ALS mice will allow us to suggest targets for treatment. The work we are doing here will hopefully extinguish the flame that is ALS and lead to restoration of hope within the patients. Thank you.
Last year, in August, a 183-mile stretch of the Yellowstone River had to be closed. The reason? A tiny parasite, only a few cells in size, wreaked havoc on the fish populations. Interestingly, this tiny parasite is an animal just like you and I. It is a non-bilaterian animal. Animals can be broadly divided into two groups: bilaterian animals like us who have bilateral symmetry, that is only one plane dividing our body into two mirror images, and non-bilaterian animals like the parasite, jellyfishes, sponges, corals who have no bilateral symmetry. These are the animals I am working with. I want to look at the mitochondria in these animals. Now, in each of our cells, mitochondria are very important organelles. They are called a powerhouse of the cell because they generate almost all of the energy our cells need. They are also involved in many important functions. In fact, dysfunctions in mitochondria have been linked to diseases like cancer, Alzheimer's, and even Parkinson's. All these important functions in mitochondria are carried out by proteins, most of which are actually synthesized outside of the mitochondria and imported into the organelle. Right now, even as I speak, there are about 1600 proteins in each of our cells, in each of the mitochondria, essentially keeping us alive. This set of proteins is called as a proteome, and mitochondrial proteomes are very well understood in some bilaterian animals like humans and mice. In my thesis, I use computational techniques to identify these mitochondrial proteins in the non-bilaterian animals, characterize their functions, and use this data to see how important mitochondrial functions have evolved in animals. Our results show that these mitochondrial proteomes in these non-bilaterian animals are very diverse with respect to size and content. Some sponges have over 800 more proteins in their mitochondria than we do, and the little parasite which shut down the river has less than half the number of proteins in its mitochondria. Another interesting thing, these proteins have a short signal which targets them into the mitochondria. In my thesis, I also analyzed these signals across the animal groups. What we saw is that these signals are actually slightly different in these non-bilaterian animals than they are in the mitochondrial proteins in us. So, with that, in conclusion, I wanted to say that mitochondria is a very important organelle and we really need to know how it has evolved, and the answer may very well lie in this group of beautiful and interesting non-bilaterian animals. Thank you.
according to the Merriam-Webster's dictionary, aging is defined as the irreversible process of reaching to an advanced stage of life. Now, with beauty and the grace of reaching to the advanced stage of life comes many health complications. Today, I am going to talk about one of that health complication which is part of my thesis, Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease that affects at least 1 to 2 individuals out of 100 over the age of 65. We already know that US is going to have as much as double population of people who are over the age of 65 by 2060. That means that we will have at least the double of number of people that are affected by Parkinson's disease. So, this is a huge health economic concerns for United States. Now, what happens in Parkinson's disease? In Parkinson's disease, a particular types of neuron called dopaminergic neurons are died in one particular parts of the brain that is called the substantia nigra, and here you can clearly see that in control you have good healthy dopaminergic neurons which are dead in Parkinson's disease patients. How do they die? One of the for surest thing we know is lot of environmental toxicants, mainly pesticides, kill these dopaminergic neurons. In a healthy cells, our neurons are protected by the surrounding cells which are called, let's call them the army. These are the army of the cells. This army of the cells actually protect this neuron from anything that comes from outside. And for so far, researchers have been only identifying what is happening with pesticides and neuron. Here is where my research comes in. I actually want to come in and talk about one of these army cells called microglial cells. When neurons are sending the sickness signals to these microglial cells come and protect the neurons. But what these pesticides are actually doing is they are keeping these microglial cells, the army of the neurons, active for very long time. And when they are active for very long time, they keep shooting the bullets. When they are shooting these bullets, it is not just killing the toxicants, it is also killing the neurons. And that is where you see the Parkinson's disease only in the later stage of your period, because when couple of neurons are dead, you will not see anything, but when more and more neurons die at the later stage of your life, you see the symptoms of the disease. So, here I come in and I identified some more proteins that are particularly activating this army of the cells and killing more neurons. And what I have found out is that some pharmacological compound actually when we give it to the animals, they actually stop the activation of this army of the cells and protect the neurons and delay the disease progression of Parkinson's disease, saving the patients. Thank you.
In recent decades in the US and around the world, there's been a real push to incorporate engineering into science education efforts. In the United States, that's most clearly seen with the next generation science standards, which have a real emphasis on engineering. And these efforts are going on at all grade levels, and I'm most interested in looking at what goes on at the elementary level. Now, in elementary schools, engineering instruction usually takes the form of engineering design challenges. So imagine that we give students a bunch of different materials and we task them with designing, let's say, a windmill that can spin as fast as they can, and you give them some time to make a plan, build a prototype, test it out, maybe go and revise their design. So in that context, what I'm really interested in studying is how the field of engineering is being portrayed to these students. That is, what sort of messages are we sending to them about what engineering is, what engineers do, and how it's different than science. These sorts of questions are very important because one of the goals of engineering education is to perhaps get students interested in the field of engineering. And if that's the case, we really want to make sure that they have an accurate understanding of what it is. So I've been working with a group of elementary teachers who have been implementing these sorts of design challenges with their students. And what I've been finding is that by and large, these teachers are interested in accurately portraying the field to their students. It's a learning goal for them. But the instruction that they actually give doesn't always reflect that goal. Most critically, what these teachers really need to do is they need to help their students connect what they're doing in the classroom to what real engineers do out there in the world. They need to engage students in conversations about the similarities between these two things, but also, perhaps more importantly, the ways in which these activities are quite a lot different than real engineering work. What I've been finding is that these conversations simply aren't happening. And the ongoing part of my research is trying to figure out why this is the case. Is it that teachers simply lack the content knowledge to have these conversations with students? Or do they lack certain pedagogical teaching skills to engage in those conversations? We need to figure this out because clearly these teachers need some support. They need some professional development, teacher education. And if we don't know where the knowledge gaps are, we don't really know how to help them. And we need to help them because if we don't, then we'll have a whole group of students who have maybe experienced a whole lot of engineering activities in their classrooms, but still have no idea what engineering really is all about. Thanks.
Well, thanks to the presenters. Let's give them a round of applause. So at this time, the three judges have what I imagine will be a difficult job. And to give them some privacy and quiet space to work, we'll escort them to a nearby area where they can confer. And when they're complete with their analysis of the presentations, they'll rejoin us and we'll make the announcement of the winners. Everyone in the room is welcome to cast a ballot for the People's Choice Award. Judy will show you the way. So in case you are unaware of the address, they're on these orange slips, which we'll have available at the front of the room if you'd like to cast a ballot. Yes?
Is there cake and coffee?
Oh, yes, there are. You can go out and, for those of us who are here rather than remote, you can go out back into the hallway and have some more cake and coffee or refreshments.
Is the People's Choice going to be a third person?
Yes, the People's Choice.
It won't be first or second place.
That's true. The People's Choice will be a separate person from the other two. That's right. Good question. How could they turn that down? Yeah, 3M tea party.
This is a little.
want some photographs from the student newspaper. Yeah, you're right.
When we get to the people.
The judges have completed their difficult job, and I'm pleased to announce the second place winner first. That person is Vivek Luana. Vivek? Congratulations.
Thank you.
And our first place winner is Alex Reed. Congratulations. And now we'll announce the People's Choice winner by having the organizing committee come up. They were the ones who had this idea, so I'd like them to take some credit for it.
Should we sit down?
Sure. Katie, would you like to make the announcement?
So People's Choice goes to Anura.
Congratulations.
Thank you so much. Thank you so much.
Congratulations.
Well, thank you all for being here. And once again, let's give a round of applause to all the presenters today. Thanks once again to our 3 judges. We really appreciate your contribution to this event. And with that, I'll just say thank you again and have a good evening. For the participants would stay, we'd like to take some photographs.
And if we could have just on the count of 3, everybody shout out, "Happy birthday" to Dean. 1, 2, 3. Happy birthday!
Thank you. Thank you.
That's why you planned to put it out.
Yeah.